Hello, everyone. This is William Grant Hale Jr. I'm here today to talk to you about receptor tyrosine kinases and cancer. In the beginning, in the introduction, today I'm going to tell you what I'm going to talk about for this period. I'm going to present the developments of the receptor tyrosine kinases as they relate to cancer. The 8 things that I'm going to talk about today are the tyrosine kinases early discoveries, epidermal growth factor receptor EGFRs, receptor tyrosine kinase RTK signal transduction, and receptor tyrosine kinase in vivo functions. To continue on and talk about the epidermal growth factor receptor deregulation in human cancer, a new discovery, HER2 new, receptor tyrosine kinase cancer therapies, and receptor tyrosine kinase anti-angiogenic therapy. Hopefully, after we get this lecture done today, you'll have a little bit more information and understand more about the receptor tyrosine kinases and cancer. In the beginning, though, the growth factor research began in about 1952 and resulted in the protein nerve growth factor NGF being purified using snake venom and mouse salivary gland extracts. After about 5 more years of research in this field, epidermal growth factor EGF was identified. It was found to stimulate proliferation of epithelial cells. And then later on in 1975, the specific binding sites for the EGF and fibroblasts were found. The concept of tyrosine phosphorylation gained further experimental support, where in the early 1980s, the epidermal growth factor receptors, the insulin receptor INSR, and the platelet-derived growth factor receptor PDGFR are protein tyrosine kinases that can be activated by their own respective ligands. Then the epidermal growth factor receptor primary structure. The molecular cloning led to the identification of the cloned DNA that encoded important physiological peptide hormones and growth factors such as insulin-like growth factor 2 IGF-2, NGF, PDGF, and transforming growth factor alpha. In 1984, collaborations isolated and characterized the clonal DNA sequence of human EFGR, which is the prototypical RTK from normal placental cells and A431 tumor cells. High levels of these similarities, though, between the EGFR peptides and avian oncogenes. It is the first time that animal oncogene with a human gene encoded a cell growth controlling membrane protein. That's what made it so important. When the complete sequence was cloned and completed, the EGFR clone DNA there was more information that was given. Truncations, deletions, mutations in the Verb beta oncogene were identified, and it continued to provide the first complete sequence of a cell surface receptor with signal generating ability, and it gave some insight into the molecular structure. Southern blot analysis also provided the application of a human A431 epidermal carcinoma cells. In spite of all this unique biological roles, though, RTKs are highly related in both structure and domain arrangement, similar to the EGFRs. Now, in the receptor tyrosine kinase RTK signal transduction, the RTK molecular mechanism presented a significant problem for research. It was found that the EGFR kinase domain of the chimeric protein was activated by the insulin binding, showing each RTK's use closely related mechanisms for signal transduction across plasma membranes. Then a bivalent ligand was found for the vascular endothelial growth factor VEGF receptor FTLI and the NFG receptor TRKA. The EGF stimulated the RAS oncoprotein to become active. The SH2 domains bind tyrosine phosphorylated peptide in vitro. Then continuing on, the tyrosine kinase RTK signal transduction. In 1990, several mammalian cytosolic factors catalyzed the exchange of RAS-bound GDP for GTP. With more thorough studies on RAS, RAF, and MEK, insights into how RTKs regulated the extracurricular signal-related kinases, ERKs. RTK was also found to activate P13K, the mediator for survival of the AKT, and the signal transducer and activator of transcription. Then we have the signals were also stimulated by the EGFRs and other RTKs. EGFR signal transactivation pathways were also found that they were able to be involved in hyperproliferated diseases which cause cancer. This established the EGFR functions as a point of the convergence for diverse signaling pathways with key biological outcomes. Continuing on, now we have the epidermal growth factor receptor deregulation in human cancer. In this, we had early on, it was found that EGFR overexpression in epithelial tumors showed that deregulated EGFR signaling had an important role in human cancers. And it also had increased stimulation of the EGFR through the autocrine growth factor loops was a common mechanism of RTK deregulation. The most prevalent mutation was found to be the EGFR8, resulting from gene rearrangement of alternative messenger RNA splicing. Impaired receptor downregulation was found to be another RTK deregulation mechanism. Oncogenic forms of the ligase CBL functions as a dominant negative mutants that prevent it from negatively regulating the RTKs. Then they had a new discovery of the HER2/neu. This complete primary structure of the putative shows high levels of homology to human EGFR related to HER2. Neu was an oncological significance because monoclonal antibodies, MABs, reverted transform effects in the NIH3T3 cells. There is also a correlation between HER2 overexpression in tumors and reduced patient survival and time to relapse. EGFR activation induces HER2 through heterodimerization. HER2 is a preferred partner for EGFR, HER3, HER4, and HER2 provides an extra recruitment mechanism of diverse intercellular signaling pathways. Receptor tyrosine kinases, cancer therapies. So we had the receptor, the HER2 gene in breast and ovarian cancers provided opportunities to develop both the target-specific cancer therapies and these humanizations of these MAB4D5 with therapeutic antibody development was then possible. Success was found where there were specific medications were found that were able to treat breast cancer patients, and it was able to target the human oncoproteins. Then the anti-HER2 monoclonal antibodies interfered with the oncogenic signal generation were developed. This breakthrough led to the colorectal cancer treatment being successful as well. The tyrosine receptor inhibitors were also developed to help small molecule anti-cancer treatments. Then we continue on with the cancers. Mutation in the ATP binding pocket of EGFR abrogates its tyrosine kinase functions and interferes with oncogenic signaling. This led to where this was also developed to help small molecule tyrosine kinase inhibitors. So there, once again, we have another small molecule treatment produced. This proved the worth of this class of compounds for selective inhibiting of cancers. Active non-receptor tyrosine kinase is expressed in chronic leukemia cells, CML, in addition to the ABL and the BCR-ABL. The RTKs, PDGFR, and KIT were found to be potentially inhibited by the imatinib. This is a new type of drug as well. Then the receptor tyrosine kinases anti-angiogenic therapy for the VEGF and its receptors have important functions in the regulation of tumor angiogenesis, proving that VEGF and VEGFR signaling are required for tumor angiogenesis. Dominant negative VEGFR2 mutants blocked the subcutaneous growth of glioblastomas, and this led to clinical anti-angiogenic therapy targeting either the ligand or the responding receptor. VEGF and VEGFR were then identified as important targets for therapeutic intervention in tumor growth. These small molecular VEGFR antagonists, such as SU5416, competitively block the ATP binding to the tyrosine kinase domain of the receptor, inhibiting tumor angiogenesis in vivo. So now that we have covered these 8 topics, and those topics once again are the tyrosine kinases early discoveries, the epidermal growth factor receptor EGFR, the receptor tyrosine kinase RTK signal transduction, the receptor tyrosine kinase in vivo functions, the epidermal growth factor receptor deregulation in human cancers, a new discovery, the HER2/neu, the receptor tyrosine kinase cancer therapies, and the receptor tyrosine kinase anti-angiogenic therapies. You should now have a better understanding of the development of receptor tyrosine kinases and cancer. Let's take a quick look at the references that I used. Here's the first 4. Notice there's one there, a textbook at the end. The top 3, though, are one is from the Current Visions on Target Enzymes for Cancer Therapy. Then we also have one from Nature, and we also have another one that is from the Nature Reviews Journal. Then the bottom one is from a textbook from Grand Canyon University uses. Then our last reference slide. Once again, we have 2 more. We have Communications Biology, once again another journal, and then the very last one is from the Journal of Immunotherapy for Cancer. So once again, I hope that I enlightened you at least a little bit. This is kind of a tough little subject to handle, but at least I give you a broad brush approach of this topic. If there's any questions, please be sure and contact me. You do have my email address at the GCU email box. Thank you very much for listening. I hope everyone has a nice day and everybody be really, really nice. Thank you.